Okay, for foliar sampling shows what nutrients are inside the plant. It indicates what plant needs are before deficiencies appear and can be used to fine-tune the fertilizer program to optimize yield and growth and plant health. And it overcomes the limitations of soil testing in a established planting, which is, again, not accurate for nitrogen or the micronutrients. The main value of soil testing in an established planting is to monitor pH. But as we saw in that pH table or graph, that it can be quite important to do so because it affects the availability of micronutrients and macronutrients. Supplies will need a paper bags, the submission form, and sufficiency tables used to interpret. And then clean plastic buckets or paper towels for washing are optional. The guidelines collect at the recommended time based on the crop development stage. And this is when plant growth and nutrients are expected to be relatively stable. Those are when the sufficiency ranges are established based on previous research, extensive research, and collect only from healthy plants of average vigor and collect randomly from within the planting. You can zigzag down the row or sample every nth plant in the row. So for instance, every 9th or 10th plant, just depending on how large the planting is. And this will help eliminate or reduce bias. Collect from only one cultivar, one variety, and then separate any, if we're fruit trees, separate any ones that are on different rootstocks, only sample from one cultivar on one rootstock, and then separate samples for young and old trees or blocks. And if we want to do a diagnostic sample, we'll take a separate sample from a section of the field that's poorly growing, where separate trees are poorly grown, and separate plants from healthy and productive plants for comparison. For the leaves, the total number varies based on the crop, but is generally enough to get a representative sample. Select only clean, healthy, and well-exposed leaves and from well-exposed shoots, and don't sample any diseased, insect, or damaged leaves. Collect from the current season's growth, and then collect from plants and shoots that are average vigor for that planting. Usually limit 1 to 2 leaves per shoot, and then 6 to 7 per plant in general. Collect the leaves with petioles attached. Petioles are the stems on the leaves. And then place in a labeled paper bag. Make sure it's labeled to indicate what the sample is if you're taking several samples to avoid confusion. Now, they should be sampled at the correct time. For example, for strawberries, for new plantings, they'll be collected around bloom up to harvest, March 15th to April 15th. And total number of leaves are going to be 60, and from the youngest fully mature, expanded mature leaves. The way to tell the difference is young leaves will be lighter green, and then the darker leaves will be changed to be mature. These two leaves right here were possibly the most recently fully mature expanded leaves. For peaches, apples, and other tree fruits, July and August, and for some of the small fruits like blueberry, into June. And this table is available from UK Hortifact 3001 fruit crop tissue analysis. So for our apples, we'll select from a shoot that's 45 degrees to 60 degrees from the ground at eye level, according to this recommendation, and then 1 or 2 leaves from the midpoint of that shoot where those hands are. Other sampling considerations, according to other recommendations or guides, include strawberries selecting only 1 leaf per plant, and for plasticulture, strawberries starting at bloom. And it's sometimes recommended to detach the petioles as you're sampling and then include those petioles in the sample bag. For blueberry, from the middle of current year's growth, like apples, and brambles, floricane types be sampled after harvest from the primicanes. Those are the new vegetative shoots. Semi-erect blackberries, this will be during late harvest. And for primocane types that are going to fruit in the fall, the sampling period is bloom to green fruit stage. Grape, ideally sampled at veraison, which is the color change of the clusters or berries, and then one leaf petiole per shoot. The leaf is disposed of, only the petioles are included in the sample submission bag. Apple, mid-canopy shoots, don't sample from spurs or leaves from spurs. And then for larger trees, this will be slightly higher and lower for smaller trees if it's mid-canopy. Peach, main season cultivars would be during the optimally during the first week of harvest. Washing samples, you only wash samples if they're dusty, dirty, or recently sprayed, because some fungicides, especially the class M's, contain minor elements, and this can affect the results. It's best to wait until right before a cover spray to sample in this case is optimal. Use clean bucket and municipal or distilled water. Don't use well water because that's a source of contamination. Add 1 to 2 drops of dish detergent in a wash bucket, then wash, swish them around, quickly move them over to the rinse bucket, and then a second or third rinse bucket to further wash them off. And if we have a large number of samples, change out the water every so often. Spread those leaves have been washed out on the paper towels to dry as single layer. Label if there's several samples to avoid confusion, and then air dry before 1 to 2 days before bagging, and then don't use heat to speed dry. Put in a labeled paper bag, don't use plastic because that'll cause molding, and then make a cheat sheet with the sample descriptions as a reminder. Fill out the lab form with any of the analysis, include the crop, sample date, the growth and the growth stage, as well as the basic, and then ship early in the week if possible to avoid sitting in shipping in a box over a long weekend. Chopping is optional to further mix those that sample, but the leaves must be very fully dry. They must be crispy to work, otherwise it'll just clump in that mixer. Analysis is standard across labs. That means they're comparable one lab to the other. The methods are based on sufficiency ranges, which is the ranges of nutrients in healthy plants at certain stages of development based on research. Critical values, which is the critical value is where the concentration of 5 to 10%, there will be a yield reduction. And that's usually the lower limit of sufficiency, or nutrient ratios are used less often. And those are just the interaction between 2 or more essential elements together. The nutrients will be macronutrients. NP and K, the adjustments is most effective when soil applied, not foliar. Plants don't take these nutrients in large enough amounts to make a difference. The mesonutrients, those ones are used in smaller amounts, calcium, magnesium, sulfur. The foliar sprays are more effective than soliar applications based on previous research and trials. The micronutrients, soil applications are generally ineffective in established orchards, so they will be foliar applied. But it's important to note that the plant availability is restricted, soil pH above 6.8, so keep an eye on the soil pH if you're noticing deficiency in any of these micronutrients. These soil samples were taken from a 4th-year gala on M26 Emla rootstock. And this is the 3rd cropping season. According to UK recommendations, phosphorus is medium, but it's getting towards the low side of medium. Same with potassium, whereas magnesium in 0 to 8-inch depth is very high and calcium is very high. Soil pH is neutral, the buffer pH is high, and sulfur and boron are, according to the lab, low, but in the soil, again, UK has not established sufficiency ranges in the soil for these two nutrients. Again, ignore the bars on this. In the 0 to 8 to 16-inch depth, phosphorus would be considered low. Same way for the potassium, according to UK sufficiency ranges. Magnesium and calcium are high as well. So from the tissue analysis that is included in this sample, nitrogen is considered adequate in this planting. In fact, for Gala, early bearing stage, according to Penn State recommendations, it might be a little bit high nitrogen. Phosphorus is adequate or normal, whereas potassium is getting to be on the low side of normal. And for plant analysis in the southern region, the sufficiency agent established then, it could actually be considered below normal or low, just depending on which sufficiency range is used. Magnesium is normal, and calcium is considered low in this case. As well. Sulfur is getting low, but is still normal. That's one to watch. Boron is also low, as well as zinc in this planting, whereas manganese and iron are sufficient, and copper is on the low side of sufficient, likely due to the high soil pH. So recommendations generated from this. We can apply potassium sulfate as a soil application, granular, 200 pounds, banded in the drip line, or water soluble. We're using that, 60 pounds sprayed or injected through the drip irrigation. What you're trying to do is with the granules overload the amount of potassium in the upper reaches of soil to try and force it downward. Water soluble is a little bit more movable in the soil, so it's likelier to get to the roots quicker. Calcium chloride foliar sprays are used, and the recommendation would be 5 pounds per acre in 6 to 8 sprays starting at first cover, which will result in 9 to 12 pounds of calcium. That'd be the intermediate range according to Penn State. And 30 to 40 pounds of that 29% calcium foliar per acre per season. For boron, 20.5% boron foliar, applying 4 pounds per acre at first bloom and petals fall. For zinc, we have two options, 10% zinc chelate, half a gallon in up to 6 sprays every 14 days, starting 1 to 2 weeks after petal fall. And or 10% zinc sulfate sprayed when dormant, but not within 30 days of an oil application to avoid crop damage. So to accurately interpret these, focus just like on the soil, on the test report nutrient values, not on the bars, and then use the sufficiency tables to interpret them. Balance this with the expected crop response and the potential for profit. So at the deficiency range, we'll likely get deficiency symptoms. As we increase to low and up to the critical value, yield will increase. Past the critical value, we'll get a 5 to 10% yield increase potentially. And at up to sufficient where it will level off. And then in high, we're going to get luxury consumption of those nutrients within the plant. We keep increasing the amount of nutrients supplied, we're going to get mild toxicity, and above that, we're likely to get toxicity visual symptoms. Stated another way in another graph, at that sufficiency point or shortly after, plant growth peaks and yield peaks, whereas afterwards, if we keep applying those nutrients, profits are going to start declining because we're spending more on nutrients, but not getting a result out of that plant. So for soil applied, it may take several years or longer to adjust because it takes time for those, especially if we're applying to the soil surface and not incorporating for those nutrients to get down to the root zone. Nitrogen is quicker, it moves quicker through the soil, but we want to apply later, no later than July to adjust for most crops. Strawberries are different because they set their flower buds in the fall. Foliar applied, we get a rapid response, but it tends to be temporary. So we need to reapply either annually or more frequently in the case of ripening disorders like calcium. And the yield response from this is possible during the current season, as in ripening disorder, or following years, which is most likely. And the delay in response is because of flower buds for next year's crop. These are perennial plants, most fruit crops. They developed the previous year. So for this year, we're setting flower buds to develop next year's crop. So fertilizing is not just for the here and now for foliar fertilization, but it's for future years. So we might not get an effect on the yield this year, but we hopefully will next year. With that, that's the end of my talk, and I'm available for questions.